Hello, my name is Jude Rafael Sariaga, and along with Joshua Martinez, we will go ahead to present this summary article about the functional SARS-CoV-2 specific immune memory persisting after mild symptoms of COVID. So the basis for this journal article is that individuals who go ahead to be infected with SARS-CoV-2 usually have mild symptoms, and uh, the basis in immunology is that once you're infected with uh, some kind of viral infection, you often uh, will have populations that will persist to carry functional immune cells that will protect them later on. And so upon having a viral infection infected by you, nearly 90% of these cells are effector cells, and they will go ahead to die as the infection progresses or no longer persist after um, having these symptoms, and only about 10% persist to uh, go ahead and create a continuous flow for antibody secretion that will help to stimulate a response when you get reinfected. So the authors wanted to focus whether or not within a set of two time intervals, usually 1 month and a 3 month period, to see if they can see the progress of COVID positive patients and see if they are able to create persistent memory lymphocytes. These tests usually vary with an analysis, so you'll see later on we'll be using either we'll see the works of ELISA or some view of ELISA with some kind of integration to see chemokine receptor levels or cell proliferation.
From the hypothesis to determine whether mild infection induces a lasting multi-layered defense, it is critical to assess the full organization of SARS-CoV-2 specific immune memory responses. As we cover each subset of immune memory cells and their responses to acute viral infection, we will discover if these multi-layered defenses do protect an individual upon re-exposure or reinfection. Persistent memory lymphocytes display hallmarks of protective antiviral immunity, including a numerically increased population of virus-specific memory B cells capable of expressing SARS-CoV-2 neutralizing antibodies. So first, it was assessed that SARS-CoV-2 specific immune responses were looked at 1 and 3 months post-symptom onset in individuals that had experienced mild COVID-19. They collected plasma and peripheral blood mononuclear cells (PBMCs) from 15 individuals recovered from mild COVID-19. The COVID-19 positive group had a median age of 47 and reported mild symptoms lasting a median of 13 days. The first blood sample, or their first visit, was drawn at least 20 days after a positive PCR test for SARS-CoV-2 and a median of 35.5 days post-symptom onset. As we continue with the methods, participants returned for a second blood draw, or their second visit, a median of 86 days post-symptom onset, so they could assess the quantity and quality of the long-lived memory populations. To the right, we see a figure labeled "Healthy controls do not have SARS-CoV-2, RBD, or spike-specific antibodies." So they took these two blood draws from the COVID-19 positive individuals and compared these samples to samples collected at two time points representing a similar sampling interval in a group of 17 healthy controls. All healthy controls were considered to have no prior SARS-CoV-2 infection based on having no detectable plasma SARS-CoV-2, RBD, or S-specific antibodies, as stated. Within these graphs, they only considered above 3 standard deviations of the mean of historical negative plasma samples drawn prior to 2020. By knowing and understanding how the immune response is initiated, it is expected by visit 1 that the primary response to be contracting and early memory populations to be generated, as viral load is cleared approximately 8 days post-symptom onset. When an acute viral infection is cleared, the majority of these highly inflammatory cells either die or become memory cells. Consistent with the resolution of the primary response, they found no difference in frequency of total monocytes or plasmacytoid dendritic cells among PBMCs between COVID-19 positive and healthy control individuals. They also found no difference in gamma, delta, or alpha beta C3 positive T cell frequencies, cell cycle status, expression of molecules associated with activation, migration, function, or proportions of various CD45RA memory T cell subsets. And finally, they found no difference in frequency of CD19 positive B cells. This demonstrates that the inflammatory response associated with acute infection had resolved by the first visit time period and the early immune memory phase had commenced.
So one of the things concluded from this paper is that mild COVID symptoms might induce the persistence of neutralizing anti-SARS-CoV-2 IgG antibodies. So to start up, in figure 1b, it illustrates this decrease in IgA and IgM levels, while levels in IgG don't decline compared to IgA and IgM from visits 1 and visits 2. So there's this kind of leveling off. It might not be that much leveled off, but a slight decrease among IgG levels, which goes to show a little persistence in outputting IgG. Another thing to note is that COVID positive patients had a reading with their plasma inhibited romaine binding domains binding to the angiotensin converting enzyme 2, which is this ACE2, significantly more than the human controls, the HC, denoted in blue. And this is done from the surrogate virus neutralizing test, this SVNT, which is basically a test to see if the antibodies form and maintain within blood plasma serum, and it shows that it readily is maintained. This is also seen in the plasma receptor binding domains inhibition, maintaining from figures F through H, where in F and in H you can see the levels kind of leveling off from visits 1 and in comparison to visits 2.
The presence of SARS-CoV-2 neutralizing antibodies 3 months post-symptom onset in CoV-2 positive individuals suggests that germinal center-derived LLPCs have formed. Germinal centers also produce long-lived MBCs that play a critical role in the formation of high-affinity antibody-secreting plasmablasts upon antigen re-exposure and can be long-lived. In order to test if mild COVID-19 induces a sustained enrichment of RBD-specific IgG memory B cells, they tested whether SARS-CoV-2 specific MBCs were formed and maintained in CoV-2 positive individuals throughout the study. They generated RBD tetramer reagents and used enrichment strategies to identify and phenotype rare RBD-specific B cells that are otherwise undetectable. They also tested specificity of the reagent in RBD immunized mice, and then used it to identify, enumerate, and phenotype rare RBD-specific B cells in PBMCs from healthy controls and COVID-19 positive individuals. From the tests that were run at first visit, RBD specific B cells were expanded in CoV-2 positive individuals in comparison with healthy controls, and their numbers in CoV-2 positive individuals increased significantly from visit 1 to visit 2. They found very few CD20 minus CD38 plus RBD specific antibody secreting cells in the blood of healthy controls or CoV-2 individuals at either time point. This supports the idea that LLPCs, likely in the bone marrow, are producing the majority of RBD-specific antibody at visit 2. The majority of RBD-specific B cells in the CoV-2 positive individuals were CD21 plus, CD27 plus classic MBCs, whereas the majority of RBD-specific B cells in healthy controls were naive. The number of RBD-specific MBCs in CoV-2 positive samples were on average significantly greater than in healthy individuals and increased even further at visit 2. These were the main findings, and these can all be related to the graphs on the left. From these findings, it demonstrates that IgG plus RBD-specific classical MBCs not only form and persist in response to mild COVID-19, but their numbers continue to increase from 1 month to 3 months post-symptom onset.
Another conclusion made by the author is that SARS-CoV-2 infection induces durable functional spike-reactive memory CD4 T cells. In Figure 3a, it illustrates the peripheral blood mononuclear cells incubated for 20 hours to enumerate the CD4 T cells. And in Figure 3b, the results suggest that the spike-specific memory CD4 T cells did not change between visits, and this also corresponds with Figure 3c, or C in this slide, and it increases the numbers of CXR5 when samples from COVID patients were re-stimulated. To measure the proliferative capacity, the authors measured the pre-sorted memory in naive subsets in response to re-stimulation. This can be seen in the frequency of cells with the proliferation die in Figure 3d. And in Figure 3e, it results in the illustration of CX3 or R3, which is associated with the proliferation of Th1 helper Ts that help to combat intracellular infections. The subset denotes TEM or TCM, which help to distinguish if it's central memory, which is the TCM, or effector memory, which is TEM, and to see whether it was greater in the re-stimulation of the samples. This figure showed both TCM, TEM, which illustrates that TEM, that some will proliferate and repopulate the memory pool upon re-encounter of the antigen spike protein. And in Figure F through I, it results in the analysis of the cytokines that help to distinguish if the overall immune response is to prioritize B cell activation or antiviral program initiation. In Figure G, it illustrates the majority of cytokines from restimulation increasing levels of interleukin 2 and interferon gamma. And in Figure H and I, it illustrates the immune system's priority to proliferate Th1 cells as to caution the possibility of reinfection. So it drives this functional and durable reactivity in CD4 Th1 cells. So as it goes.
Because SARS-CoV-2, RBD-specific MBC, and S-specific CD4 plus CTFH cells were enriched in CoV-2 positive individuals after 3 months, they questioned whether these MBCs had the potential to produce neutralizing antibodies if reactivated by secondary infection. To test this, they index sorted single RBD-specific B cells and sequenced the RBCRs from 3 CoV-2 positive individuals at both time points. They sequenced all IgG positive RBD-specific classical MBCs, clonal paired heavy and light chain sequences that were obtained, and expressed them as IgG1 monoclonal antibodies. This comprised 19 antibodies from visit 1 and 16 from visit 2. The figure on the left represents these antibodies being first expressed in small-scale cultures for screening and were assessed for antibody expression by IgG ELISA and specificity by RBD ELISA, where all but one antibody at each point showed strong binding to RBD. The figure on the right shows that these antibodies were then expressed on a larger scale and purified for more detailed analysis, and the specificity of the purified antibodies for RBD was again confirmed by ELISA. This set of antibodies utilized a wide variety of heavy and light chains, had all undergone somatic hypermutation, and were all unique clones. Somatic hypermutation levels in both heavy and light chains increased on average between visit 1 and visit 2, but these differences were not statistically significant. By the data, it is determined that RBD-specific MBCs induced by SARS-CoV-2 infection are capable of producing a variety of neutralizing antibodies against the virus and could contribute to protection from a second exposure.
Figure 5 goes to illustrate the summation of the following components of analysis throughout this paper. This was shown as a heat map indicating red as activity above the metric mean and blue as the threshold for the significant response. In all but one of the COVID patient samples had higher IgG antibodies, improved plasma inhibition to the ACE2, and as to reiterate, ACE2 is usually seen in inflammation. It's more of a harmful kind of enzyme than it is an aided for immune responses. Also within this result was within the 14 positive COVID positive patients were higher numbers of memory B cells, as well as an increase in effector cytokines compared to that of the human controls. So we see express the activity in all of the COVID positive patients except for the one, which I believe is the one denoted as COVID positive patient 3. So
to conclude the author's data, they found that 3 months after mildly symptomatic COVID-19, recovered individuals had formed an expanded assortment of SARS-CoV-2 specific immune memory cells that exhibited protective antiviral functions. They also showed that recovered individuals formed a multifaceted defense and that it increased month by month. They demonstrated elevated IgG plus RBD-specific plasma antibodies and neutralizing plasma are generated and maintained at elevated levels for at least 3 months post-SARS-CoV-2 infection. This helps advance our knowledge of COVID-19 and the immune response to it by understanding that if natural infection induces viral-specific immunological memory, then that could influence how we prevent transmission and treat the disease severity to control the virus. The next steps would be to understand what level of antibody is needed to contribute to protect and whether that level will be maintained in the long term. But in terms of treatment, although recovered individuals experience short-term immunity and form a viral-specific immunological memory response, it is suggested that a virus vaccine with a specific isolated antibody would be similarly successful and functional in eliciting a stronger response.